Welcome back to my channel. Here in this video, I am going to discuss about the process of microbial strain improvement. In my previous video, I have described some methods called screening to isolate and identify industrially important microbes from different natural habitats. Now come to the next problem. You have isolated one microbes using screening methods, but it produces very low amount of antibiotic that is not profitable and also not viable for fermentation, or the microbes can able to utilize very low cost plant-based starting materials but not able to tolerate more than 10% final alcohol concentration in the fermenter. Or you have isolated one new strain that can able to produce penicillin in good amount than any other strain isolated before. But here again your strain changes its genetic composition very rapidly during storage or after 4 to 5 times passage in the growth medium. So what to do next? All you need to do is to change the genetic composition of the microbes. This process of enhancing the biosynthetic capabilities of microbes to produce desired product in higher quantities is called microbial strain improvement. Here is the list of targets for strain improvement. Generally, microbiologist wants to improve the strain to increase the growth rate. More growth means more amount of final product, or to stabilize the genetic material in those microorganisms that change it more frequently, or try to increase the cell size so that it can be easily removed from the culture medium at the end of the fermentation. If the size of the microbes are large, they can be easily settled down at the bottom of the tank due to gravitational force or low speed centrifugal force. Strain improvement also applied to change the fermentation type, like from solid state to submerged fermentation. It is also eliminate feedback inhibition and block toxic byproduct formation as much as possible. Now let's take some common methods that are applied for the strain improvement. Strain improvement technique can be divided into two types based on whether microbes uptake any foreign DNA or not. The traditional method is called induced mutation. Here no foreign DNA material is inserted into the microbial genome. Nitrogenous base sequence of the microbial gene are altered upon exposure of mutagenic agent. When exposed to mutagen, mutations introduced randomly into the genetic material. After the exposure, altered phenotypes are selected for further analysis. Sometimes the mutated strain is again exposed to mutagenic agent until it gives a better result than wild-type phenotype. For example, 21 rounds of mutation and selection were carried out at different laboratories on Penicillium chrysogenum, and at the end, it can able to produce 55 times more penicillin than its parental strain. Some commonly used mutagens are mentioned in this table. Among them, UV radiation and alkylating agents are more commonly used mutagen. Scientists have used both radiation and chemical mutagen on Penicillium chrysogenum. As a result, penicillin became the very first commercial antibiotic. Now the question is, which mutagen will be the best for my strain? All mutagenic agents available in the market may not equally effective for all organisms. So you should try most of the mutagenic agents on your target microbe and check which one is more effective. Not only that, you have to check some other parameters before choosing a particular mutagen, like the safety of the mutagen, because many mutagens are carcinogenic in nature. The process is simple or complex, and the availability of the necessary equipment and chemicals. Among physical agents, UV radiation is the preferred because it doesn't require much equipment and it is relatively effective against most of the microbes. Chemical methods other than NTG can be used in combination with UV to speed up the process. Now the question is, after exposure of the mutagenic agent, how do we know that the genetic composition of the microbes is changed or not? Because classical mutagenesis is a random process and sometimes mutation occurred at different position. Generally, the selection of the mutant is based on the changes in morphology or color, nutritional requirements, and by screening. Although the exact mechanism behind this phenomenon is unknown, but it has been observed that when morphology of the strain changed, it shows higher production of the metabolite. For example, When Gibberella species is exposed to UV rays, it shows two mutant phenotypes, different mycelia and soluble pigmentation. It was observed that the unpigmented morphological mutant able to produce more amount of gibberellic acid. One or more than one metabolic pathway is actually responsible for the product formation. Microorganisms usually have some regulatory mechanism that actually control the amount of metabolite production, called feedback inhibition. When the concentration of the final product increase, it inhibits the very first step of the metabolic pathway by itself. Therefore, microbes stop metabolite synthesis after a certain point. Until or unless the regulatory mechanism is suppressed, the strain will not produce more amount of product. Microbial culture, which have multivalent mechanism of regulation like concerted repression or feedback inhibition, are generally used for strain improvement. Mutants which have lost the ability to synthesize one of the end product capable of feedback inhibition or repression are selected for strain improvement. For example, glucose is converted to aspartic semialdehyde, which further converted to lysine, methionine or threonine depending on the requirement. The end product lysine and threonine both can inhibit the activity of the very first enzyme, that is aspartate kinase. As a result, the microbes stop producing all three amino acids. A mutant defective in threonine and methionine production is selected, which produce more amount of lysine than parental type. Like substrate analog can also interact with the regulatory site of the enzyme. As a result, they can also able to regulate the metabolic pathway. Binding of the substrate analog at the regulatory site of the enzyme inactivate the enzyme. The effect is toxic to the cell. The toxicity eliminates all sensitive mutant cells in the population. In this screening process, microbes will survive if they can tolerate high concentration of substrate analog. For example, alpha amino beta hydroxyl valeric acid is the substrate analog of threonine. Selection of mutant strain that can able to tolerate high concentration of substrate analog is done in two stages. The analog of threonine is added during the preparation of nutrient agar and poured onto the sterile petri plate and allowed it to solidify in this way. A second layer of the same medium without analog is poured onto it and allowed it to set. After some time, diffusion of an analog into the upper layer of the medium takes place. As a result, a concentration gradient is developed at the surface in this direction. Now, a culture previously treated with mutagen is sprayed onto the surface of the medium and selection of any mutants offering resistance to high concentration is conducted. Our last method for screening of mutant phenotype is based on the ability of the microbes to produce high amount of end product. Generally, it is done by Agar-Zone diffusion method. Let's check one example. Methylene blue Agar-Zone assay has been used for mutant screening and selection of Streptomyces species. If the color of the dye changes surrounding the colony, that means it produces amino acid lysine. If the diameter of the zone increase after the exposure of the mutagenic agent, that means the species able to produce more amount of L-lysine. Although the classical mutagenesis methods are used more frequently than any other methods, but it has some limitations. Like it is non-specific and time-consuming. It requires knowledge of the gene that control the target product and the genetic map of the organism. To overcome those problems, site-directed mutagenesis and in vitro mutagenesis are used in combination with genetic engineering to modify gene of interest or part of the gene. Now I will discuss about the next one, that is the foreign DNA is inserted into the bacterial genome. Among them, the most widely used modern technique is called Recombinant DNA Technology. Various new tools and strategies have been developed in the past few decades like cloning, transgene production, protoplast fusion, etc. With the help of RDT, now it is possible to transfer specific gene from one organism to another, no matter whether they are closely related or not. In contrast to other process, modern recombinant DNA technology has some advantages like unlimited opportunity for the production of novel combinations of gene are there. It is highly specific and well controlled. This can be used to increase the product yield by removing metabolic bottleneck or by amplifying or modifying specific metabolic steps. Totally new capabilities can be inserted into the industrial microorganism. Microorganism can be manipulated in such a way so that it can able to produce novel compounds, or it can able to use cheap and complex substrates. As there is no restriction to the origin of the gene, now microbes can able to produce plant as well as animal proteins. Now let's take one example of the potential application of RDT in the field of industrial microbiology. Traditional chemical synthesis of biotin is costly and not profitable. When biotin operon is inserted in a multi-copy plasmid present in the E. coli cell, can increase biotin production up to 10,000 times than wild-type E. coli cell. RDT gives economic advantage over traditional chemical synthesis. Not only that, you can check the advantages of RDT in industrial microbiology in this table. The references are also provided for those who are really interested to know in details. Although RDT has several advantages than traditional mutagenesis, but this method have not totally replaced the traditional mutagenesis, and the two approaches should be viewed as a complementary strategies for strain improvement. Now we will talk about the role of metabolic engineering for strain improvement. The improvement of cellular activities by manipulation of enzymatic, transport and regulatory functions of the cells with the use of recombinant DNA technology is called metabolic engineering. This method requires overexpression or downregulation of certain protein that has some important role in the target metabolic pathway. As a result, cell converted into an efficient chemical factories for the production of chemicals, fuels and pharmaceutical products. Information gathered from laboratory experiment are used for the construction of computer model that are used to understand basic cellular mechanism to achieve desired goals. The clear knowledge of the enzymes involved in the metabolic pathway is necessary for metabolic engineering, as well as how the pathway is regulated and how substrate and end products are transported are also required. As more and more amount of informations are made available, it will become easy to construct computational model. Metabolic engineering can be divided into three types based on the approach. Classical metabolic engineering depends on RDT for the changes in the cellular metabolism. It can be achieved by changing its pathway enzyme or regulatory proteins. Inverse metabolic engineering takes advantage of the knowledge learned from the mutants or existing strain with improved performance. In vitro metabolic engineering, all metabolic components are assembled like purified or partially purified enzymes, non-enzyme components such as cofactor, and then without utilizing a cell, a pathway is designed to convert a certain substrate into a target compounds. At last, I will describe one application of metabolic engineering in alcohol fermentation. Yeast generally ferment glucose to ethanol, but it can't able to utilize lignocellulose or hemicellulose biomass as starting material for alcohol fermentation. Both starting materials are cheap and abundant, and most important of all, contain glucose. Some microbes can able to utilize those cheap carbon source via homoethanol pathway. Yeast cell can be improved by the introduction of additional sugar utilization pathway so that it can able to utilize a wide spectrum of sugar as a starting material for ethanol fermentation. By adding those genes inside the yeast cell, not only improve its performance but also convert it into a microbial biocatalyst that can use cost-effective growth medium. Although the whole process of metabolic engineering is standardized and conceptualized, but many regulatory control mechanisms in nature are not fully understood yet. Metabolic engineering is time consuming and efficiency is very low, and it is costly. Metabolic engineering still requires further development before its full potential can be fully realized. So that's all for now. I hope you enjoys this topic. Share your opinion in the comment section and subscribe my channel if you like my videos. And finally, thanks for watching.